Let us pass on to the next phenomenon, crossing over. Crossing over is nothing, it is a recombination of genes due to exchange of genetic material between homologous chromosomes. It is a mutual exchange of segments of genetic material between non-sister chromatids of two homologous chromosomes to produce recombinations or new combinations of genes. The non-sister chromatids in which exchange of segments have occurred are called recombinants or crossovers, while the other chromatids in which crossing over has not taken place are known as parental chromatids or non-crossovers. Then, an example cited by C.B. Hutchinson. C.B. Hutchinson in 1922 crossed a pure breeding colored and smooth grained maize variety with pure breeding colorless and shrunken grained maize variety. Plants of F1 generation, that is first filial generation, possessed colored and smooth grains, showing that both the expressions are dominant. Expressions are dominant. F1 plants were obviously heterozygous for the two traits. F1 plants were then test crossed with double recessive parents, and it resulted in 4 types of offspring in the ratio of 27:1:1:1:27 instead of 1:1:1:1 ratio expected for independent assortment. And the above ratio is possible when the genes of the two traits do not show independent assortment. Apparently, the two genes are linked in the same chromosome, but 1.8% of the non-sister chromatids of the homologous chromosomes show crossing over in between the two genes by exchanging segments. And next one is recombination and crossing over. A new grouping of genes or a new combination of characters which is different from the parental types is called recombination or recombinant type. It is produced during crossing over that occurs during meiosis prior to gamete formation. This recombination is produced due to crossing over that occurs during meiosis before gamete formation. The frequency of recombination is directly related to the frequency of crossing over. However, sometimes 2 or even more crossing overs may occur simultaneously in the same non-sister chromatids of the homologous chromosomes without altering or without changing the frequency of recombination. Therefore, frequency of crossing over may be higher than the frequency of observed recombinations. And the frequency of recombinations or crossover value, it is represented by COV, is calculated by using the formula number of recombinants divided by total number of progeny into 100. Then this COV between genes of red eye and normal wings means that genes are located 10.7 units apart on the same chromosome. In the given cited example, the frequency of recombination is 10.7%. In the given example, the frequency of recombination is 10.7%. We calculated this with the help of the formula, that is number of recombinants divided by total number of progeny into 100. We get 10.7%. So this COV or recombinant frequency of recombination of 10.7% between the genes of red eye and the normal wings means that these genes are located 10.7 units apart on the same chromosome. Then the factors influencing crossing over. There are different factors which influences the crossing over as well as also linkage. It is also related to linkage. First one, distance. The main factor is distance. The physical distance between the two genes determines both the strength of the linkage and the frequency of the crossing over between two genes. The strength of the linkage increases with the closeness of the two genes. On the other hand, the frequency of crossing over increases with the increase in the physical distance between the two genes. And the second factor is age. Increase in age decrease the degree of crossing over in most of the cases. Then sex. Male Drosophila shows little crossing over, and the phenomenon of crossing over is quite common in the female fly. Negligible crossing over is also reported in one sex of some other heterogametic organisms. Then fourth one, X-rays. Exposure to X-rays increases the incidence of crossing over. Whittinghill produced a number of crossovers in male Drosophila with the help of X-rays. Then fifth factor is temperature. Variations in temperature increase the frequency of crossing over. Then another one is heterochromatin. Presence of centromere and heterochromatic areas, that is near telomere, decrease the rate of crossing over. Then chemicals. A number of chemicals present in the food have been found to change the degree of crossing over in animals. Then interference. One crossover reduces the occurrence of another crossing over in its vicinity. The phenomenon is called interference, and the coincidence is the ratio of observed double crossover in relation to expected double crossover on the basis of non-inheritance or independent occurrence. Coincidence is small when interference is high. So, the coincidence will be very small when the interference is high. Then, some of the importance of crossing over. Crossing over is a means of introducing new combination of genes and hence traits. It increases variability, which is useful for natural selection under changed environment. Since the frequency of crossing over depends upon the distance between the two genes, the phenomenon is used for prepare linkage chromosome maps, and it has proved that genes lie in a linear fashion in the chromosome. Breeders have to select small or large populations for obtaining the required crossovers. For obtaining crossovers between closely linked genes, a very large population is required. Useful recombination produced by crossing over are picked up by breeders to develop useful new varieties of crop plants and animals. Green revolution has been achieved in India due to this selective picking of useful recombinations. Operation Flood or White Revolution is also being carried out on the similar lines. Let us have a look at types of crossing over. Types of crossing over. Crossing over can be single, double or multiple. Single crossing over. Crossing over occurs at one point between two non-sister chromatids of a homologous chromosome pair, and there are two parental types and two recombinants. When in the case of double crossing over occurs at 2 points in a homologous pair of the chromosomes. And double crossing over is again classified into two: reciprocal double crossing over and complementary crossing over. Reciprocal means 2 points of crossing over occur between the same non-sister chromatids. Then the complementary crossing over means the two crossing overs involve 3 or all the 4 chromatids, so that the number of crossovers is 3 or 4 with occurrence of one or no parental type. Then third one is multiple crossing over. There are 3 or more points of crossing over occur in the same homologous chromosomes. Double crossovers and parental types may or may not occur. Then when come to the mechanism of crossing over, chromosomes not get replicated in the S phase of interphase. Interphase means resting stage. Therefore, The leptotin chromosomes are double-stranded, though the two strands are not visible due to presence of nucleoprotein complex in between the chromatids. First one, synapsis. Replicated but apparently single homologous chromosomes come to lie side by side with similar gene loci of two chromosomes exactly opposite. They come to lie side by side. It occurs in the zygotine stage of prophase 1 and the phenomenon is known as synapsis. So, the synapsed pairs of homologous chromosomes are called bivalents, and the small amount of unreplicated chromosome, that is about very mere percentage, 0.3 percentage, if present, also undergoes replication. The two homologous chromosomes are held together by a synaptimal complex. Then next one is crossing over. It occurs in the packaging stage at 4-strand stage with the help of enzymes endonuclease, exonuclease, R protein and recombinase. There is breakage of chromatid segments, exchange of non-sister chromatid segments, and later their fusion in new places. And third mechanism is tetrad stage. Here the synaptimal complex begins to dissolve except in the region of crossing over. So, except in the region of crossing over, that synaptimal complex begins to dissolve. Therefore, chromosomes separate and chromatids become distinct at most of the places. As a bivalent appears to have 4 chromatids, now it is known as tetrad stage, and it occurs in the diplotin stage of prophase 1. The synaptimal attachment points between the homologous chromosomes are called chiasmata. In later stages, this chiasmata tend to shift to the sides, and this phenomenon is known as terminalization. As the chiasmata tend to shift to the sides, it is known as terminalization. Many of them disappear prior to metaphase 1.